Inwardly rectifying potassium channels represent another important and very large group of potassium channels. As we've already discussed, inwardly rectifying potassium channels differ in an important respect to other types of voltage-gated channels in that the open probability of inwardly rectifying potassium channels increases as cells are hyperpolarized rather than depolarized. These channels differ from other types of voltage-gated channels in another important way as well, and that is that their gating mechanism differs quite substantially. In most voltage-gated channels, a voltage sensor is expressed as part of the channel protein. In inwardly rectifying channels, in contrast, there's no voltage sensor in the protein itself. Rather, gating occurs as various positively charged ions within the cytoplasm block the inside of the channel pore, the inward mouth of the channel pore. The most important of these intracellular ions are polyamines, which are positively charged long-chain aliphatic compounds, and the most potent of these is spermine, which has a charge of +4. Also spermidine, having a charge of +3, and putrescine play an important role in the gating of inwardly rectifying channels. Magnesium ions, which of course have a charge of +2, also can block the channel pore and gate the channel in this manner. The way the gating works is the following. When the membrane potential of a neuron is very negative, the positively charged ions are attracted towards the cytoplasm away from the membrane, thus removing the block of these ions from the channel pore. In contrast, when the channel depolarizes, these ions are forced into the inner mouth of the pore, blocking the channel, and the open probability decreases. The binding site for these ions is within the transmembrane electric field, within the membrane itself. So there are binding sites for these cations within the channel pore inside the membrane region of the channel. Inwardly rectifying channels come in many different types, and some are strongly rectifying, others are weakly rectifying. The strong rectifiers have a very low open probability when cells are depolarized, and weak rectifiers, the open probability varies very little as the voltage is changed. The figures on this slide show voltage clamp records of strongly rectifying and weakly rectifying, inwardly rectifying potassium channels. In this diagram on the left, we are seeing voltage clamp records from a strong inward rectifier. When cells are hyperpolarized, this is the command voltage shown here, cells are hyperpolarized, large inward currents are generated as potassium flows into the cell because the voltage is more negative than the potassium equilibrium potential. In contrast, when the neuron is depolarized, as is shown here, very small currents are evoked, demonstrating that the open probability of these channels is very low. We can plot these results on a current voltage plot, and that's shown here in green in the this middle diagram, where for negative voltages, hyperpolarized voltages, there's a large current, meaning that the channel is open, and for positive voltages, depolarizations, there's a very small current, showing that the open probability is very small. This is what we mean by strong rectification, that very large currents are seen for hyperpolarizations and very small currents are seen for depolarizations. Of course, this rectification is opposite of what we'd see for a delayed rectifier potassium channel or other voltage-gated channels, where the open probability would be high for depolarization and low for hyperpolarization. The voltage clamp records for a weak rectifier are shown here. The second set of traces on the left, where we see that for both hyperpolarizations and depolarizations we record significant currents, and this is plotted here in the blue trace, where we see that the slope of the curve and thus the conductance changes very little between hyperpolarized voltages and depolarized voltages. The IV curve on the right demonstrates that magnesium and spermidine can gate the channel and are responsible for the rectification properties of the channel. These records were made from a inside out patch, so that the inside of the channel pore is exposed to the bath solution, so that the ion concentrations could be changed by the experimenter. Now, when there's no divalent or multivalent cations present in the bath solution, that is, there's no magnesium or spermidine, we see that the current voltage relation for this inward rectifier channel is linear. There's no rectification at all. However, if we add either magnesium or spermidine to the bath solution, then when the cell is depolarized, the cations can block the pore, the inside face of the pore, and open probability drops significantly at these depolarized voltages. So either magnesium or spermidine can function to gate the channel and produce this rectification. There are several different types of inwardly rectifying potassium channels, and I've listed them here according to the mechanisms by which they are gated. KIR4 channels, this of course stands for inwardly rectifying potassium channels, type 4, are modulated primarily by the membrane potential, as we saw in the last slide, and these channels are expressed in very high numbers in astrocytes, the principal type of glial cell in the brain, and represents the principal potassium conductance of glial cells. KIR6 channels are ATP sensitive channels, and with increasing concentrations of intracellular ATP, the channel closes. These are the so-called K-ATP channels, and they are expressed at high density in the beta cells of the pancreas, among other cells in the body, including neurons in the brain. And it's quite interesting how they regulate insulin secretion in beta cells. When glucose levels in the blood go up after you eat a meal, this leads to a transport of glucose into the beta cells. This in turn raises intracellular ATP levels. The K-ATP channels, these inwardly rectifying channels that are sensitive to ATP, then close, and this leads to a depolarization of the cell. The depolarization in turn leads to calcium increases and a secretion of insulin. So these channels play a direct role in insulin secretion and regulation in the body. KIR3 channels are G-protein coupled channels. They are modulated by G-proteins. They are so-called GERK channels, or G-protein coupled inwardly rectifying potassium channels, and they are directly modulated by the beta-gamma subunits of G-proteins, and with the change in open probability of these channels, changes in neuronal excitability are seen. KIR1 channels are pH sensitive. They're regulated by protein levels, proton levels. One of these channels is called the ROMK channel, which is pH sensitive. Now, inwardly rectifying potassium channels have many functions, and I'll just mention a few here. The channels contribute to the generation of the resting membrane potential in neurons, glial cells, and muscle cells. These channels also modulate neuronal excitability, and as I've already mentioned, they are the primary conductants in astrocytes, and as such they contribute to potassium homeostasis in the brain, which is largely regulated by potassium levels, are largely regulated by glial cells in the brain. Based on what we now know about the gating mechanisms of inwardly rectifying potassium channels, I'd like you to think about the following exercise. And what I'd like you to do, and what we'll be doing in class, is to draw the current voltage relation of a channel that has the following properties. The channel is permeable to both sodium and potassium, so this differs from inwardly rectifying potassium channels, which are permeable only to potassium. However, like the inwardly rectifying potassium channels, the KIR channels, the channel pore is blocked by magnesium. However, the magnesium binds to and blocks the extracellular end of the pore instead of the intracellular end.